Hi, everyone. Welcome to the Spatial Omics Seminar Series 6. Today, we are very excited to have Dr. Vlachos from Boston area. And briefly, Dr. Vlachos received his PhD from Greece. And then after that, in 2016, he moved to Broad Institute of MIT and Harvard, where he worked on translational and precision medicine, especially his focus was in non-coding variant fine mapping and characterization, typically using in silico models. Then after his experience at the Broad Institute, he moved 4 blocks, as he just explained to us, to start his lab at the Beth Israel Deacon's Medical Center as a faculty. And first, he started as a director of bioinformatics precision RNA medicine core at the Harvard Initiative for RNA Medicine. Also a faculty at the Department of Pathology, co-director of the bioinformatics program at the Cancer Research Institute. And recently, he has initiated this effort called Director of Spatial Technologies Unit, for short STU. And he's going to be more giving details about this initiative to our community today. And he'll be presenting his work on understanding the COVID-19 cellular phenotypes across tissues using single cell and spatial technologies. And without further ado, I'd like to turn over to Dr. Vlachos.
Thank you so much. So first of all, I cannot see myself. So if I'm out of the camera, just wave or tell me something. So because something happened to my Zoom window, but it's okay. I would like first of all to thank you, and sorry, let me turn off my phone. The organizers for this amazing seminar series, for putting the time and the effort to bring the community together, and absolutely thrilled to be here with you today. So all right, my lab, as was mentioned, is located in Boston, and we focus on the integration and application of novel technologies, data-driven approaches for precision medicine applications. So we span from basic to translational research. Located in Boston, most of the footprint is in BIDMC, HMS, and the Broad. And our research program is quite diverse. This is the lab is, as I was saying, 80-20. Dry, it's a humid lab, but it's mostly dry. That gives us the ability to shift between topics and create a comprehensive program. There are some things that are overarching, a comprehensive approach that takes into account coding and uncoding genes, self non-self recognition. We have a newly minted arm with microbiome work, and also, as I mentioned, it goes into translation, but AI ML from the very beginning to bring the data together, and also bulk for the past decade, single cell for maybe 5 years, and spatial during the past 2 years are what's really driving the limits of our science. The limits of your technology usually dictate how far you can go. So in our translational research arm now, we usually develop technologies that integrate multiple components for precision medicine applications. And I will move very quickly. We really love bulk sequencing. It has been a major workhorse since 2011. And now, it's 2021, so 10 years after, it enables us to, apart from gene expression, of course, to extract multiple layers of information, coding, uncoding genes, coding, uncoding variants, TCRs, BCRs, now even RNA processing, and now we have a paper under review. I hope it will go well in collaboration with Rob Patro's group and Joe Paulson for extracting microbial component in high accuracy. So bulk sequencing is amazing. Data are readily available. It's cost effective. It's a very rich modality. The methods for data analysis are very robust thanks to tremendous efforts from the community. it's a very mature platform and it's continuously improving. So, however, there's always a twist. Despite the incredible derived information, it's still bulk. And the lack of granularity is that imposes limitations to the questions that you can ask. And in the spatial seminar, there should be a smoothie showing up somewhere. And we know that tissue architecture is very informative. Pathology or basic research, we have people looking at tissues for decades or more than 100 years. So we know how important architecture, localization, microenvironment are. And now we have stopped discussing about cell types. During the past, I would say, decade, we're discussing about cell states and cell populations. And as we see here, in this slide, which is rather recent, we see all the different stages for CD8 T cell differentiation. But if you go into single cell assays, you see that each of those stages actually has substates and subpopulations, and the granularity can further and further increase. So bulk for these types of questions can definitely not cut it. And of course, single cell came and provided the opportunity to interrogate independent transcriptomes or multimodal now with multi-omic information, but you still lack the topology, you still lack the localization, you still lack the in situ information. This is where this is where spatial comes with a promise of bringing everything, but keeping the tissue information there. Yeah, and as I was saying to Ahmet, this seminar series gives us a little bit of time to go beyond what we usually present in a conference and discuss a little bit about strategy and the field and the challenges, but also how we believe things will move on and what we're doing about it. So of course it's an opportunity, and there are also inherent challenges. First of all, there is a consensus, and I don't think anybody disagrees, that spatial techniques are important, revolutionary, and they're here to stay. They were named Nature Methods Method of the Year 2020. And I would like to remind that single cell technologies, which still feel extremely new, they were Nature Methods Method of the Year 2013. So this domain is very, very new, and new comes with promises, but also with challenges. If we have an overview of the technologies, and the first thing we think is about the technology, where the technology is, what the methods are, and you can see the tremendous advancements during this seminar series every Friday. But you can see, because this is so new, there is still fragmentation. There is no, and I don't know whether there will be, but there is no single instrument that does like absolutely everything, or a single assay. And we usually focus on the resolution. Some are super res, subcellular resolution, cellular resolution, region of interest resolution, and then it's the capture, the plexity from a couple of genes or dozens of genes or proteins to hundreds to thousands to whole transcriptome assays. But if you actually try to apply these methods, not just for the methods development, but if you want to put them in a research or diagnostic setting, you understand that there are differences that go way beyond that. Of course, the first thing we think is the cost, but also applicability is important. Some methods are only frozen compatible or FFP or both, and that affects really how much archival tissue you could use, or some capture proteins or RNA, or might need a lot of time from sample to results, and that could be from days to weeks to months, or might need a little bit or a lot of bioinformatics expertise. Section dimensions, is it for a very small region or is it a whole slide? So all those affect what questions you can answer with a specific technology. So even if you invest on a single instrument, matching the right question to the right technology, I think is key. And the second is, I have been asked many times about the importance of spatial technologies, and I'm very open. So this meeting is being recorded, so if I'm wrong, it might not age nicely. But I really do believe that spatial technologies will be extremely transformative, much more than single cell. Even we love single cell, we do single cell, And I believe that it will rival or most probably surpass bulk. I know it's a very big statement, and happy to explain further how I reached this conclusion during the Q&A if there is interest. But there are three quite prominent challenges. Method development is definitely the obvious one. And there are many ways we can improve, but I feel that this is coming. It's the only part that I'm 100% certain that it improves, and we see this almost every week, that there is a big advancement. The resolution will come, the plexity will come, all of these will come. Access to these technologies, but not access, there is an instrument that will use it, but access that can scale, that can really be embedded in everything we do, as RNA-seq, for instance, is now. That is a different problem to be solved. And it's not just purchasing an instrument or setting up an assay. Scalability, access to expertise, training, education from junior to senior scientists, but also stakeholders, opinion makers, time and processes from sample to data. It cannot be months. It's a big issue. Research grade and clinical grade applications. What do we need to make it into the DX world? What do we need to do to CLIA certify? What are the next steps? And data analysis is already a bigger roadblock than data generation. It already is, and I think it will become bigger and bigger. For data that we generate in a month, it might take us multiple times that amount of time to reach conclusions. So if we really want spatial to be embedded in everything we do, we need an equal balance and a lot of effort in all these frontiers. So, and I have this twice. Standardization will make, and best practice will make the field mature. And this is a community effort as the seminar, but also we have to get together for the boring parts as well, which is, for instance, standardizing how we approach things. So to this end, with two great colleagues, Frank Slack and Win Hyde from BIDMC, we established the BIDMC Spatial Technologies Unit with tremendous support from the state of Massachusetts, the host institution, Ahmet Coskun Initiative for RNA Medicine. And the goal was to create a space where different technologies can be side by side, and this we see that in the seminar series, but also can be integrated. And it faces as if outwards facing facility, which means first come, first served. It's not about the host institution, and the goal is to make these technologies accessible to the community. And so working together with the technology developers, there are multiple assays I will discuss a little bit more, but there is equal focus on training, on healthcare applications, and on new technology rollout. The technologies, Ahmed, you asked me that are currently rolled out, and they're right here, right now, and can be accessible. They capture quite a wide spectrum of the plot that I showed. Ultra-res, subcellular resolution using the MERFISH/MERScope, cellular resolution, multiplex immunoassays, either high throughput or lower throughput, but higher plexity. Spatial transcriptomics from the 10x Visium and the nanostring genomics, but also scalability has been a major strategic decision, and the ability to do more samples, and also supportive equipment like large-scale tissue microarray that can reduce costs, or screen a high number of samples, or autostainers and imaging. So having all those in the same space enables you to just open the door, get the sample, and from the same tissue sample to automatically dissociate, and then using the robotic system to get single cell, or from frozen tissue dissociate, and then do automated single nuclei suspension and sequencing. Of course, frozen tissue can be sectioned. We can section in-house in the same space, auto stain, and then do either multiplex immunoassays with few or many targets, or single molecule fish, or using the genomics platform, region of interest based, or spot based transcriptomics. in the same location. So that makes a very seamless environment. And for many of those technologies, we have also established the same protocols for FFPE, and we're working with technology developers to fill the whole spectrum. So today I'm going to focus a little bit about our work using the GMX WTA, but it's just an example of how these technologies can be together. So an important aspect that I would like to stress is that to maximize the capabilities of spatial technologies as they are right here, right now, optimal integration with single cell data is key. And this is regardless of the method, whether you're doing MERFISH, Visium, GeoMix, or protein-based assays, bringing in single cell from the same samples, or at least concordant samples, can give you, increase your granularity if that's what the assay is missing, or increase your capture rate, give you multi-omic support. And I cannot stress enough that single cell have a lot of robust methods for data analysis that can give you some anchor points to understand where you're going with your spatial. And of course, for spatial, you get things you could never get from single cell: architecture, localization, your morphology, and structure function. So these are the domains that the lab is applying these approaches. And of course, the STU has a much wider portfolio. So as a title today, I would like to provide the use case of this framework using work on COVID-19 as an example. And we know a lot now about this formidable foe. Unfortunately, we're still learning, and there is more to learn. But despite the incredible progress, there are still numerous open challenges, and and especially for its tissue phenotype. But I would like to ask you to turn the clock back a year and a half ago, in the beginning of spring 2020, around March or April, where the Northeast was truly a battlefield. So back then, we have established this framework that I was discussing, spatial plus single cell, for our back then pre-pandemic mundane work. But we quickly restructured it to apply it for COVID-19, which had hit the region quite hard. And we put a goal to capture how COVID-19 affects the cellular phenotypes across tissues. So this is actually a question that was phrased very early on. This is the slide that we use in our pathology faculty meeting to present our approach for this project. And as I mentioned, this framework relies on the integration of these two modalities and how they can complement each other, and how you can get the granular component and the architectural component from both, but bringing them together maximizes the ability for both. You don't use markers to localize your cells, and you don't just deconvolve blindly what is there or try to guess. So we started this effort with Yuri and Gordon Jang from medicine, and Jonathan Heck from who is actually the head of our autopsy program. And it was a heroic effort to be able to give us the samples during an active pandemic. And that effort quickly grew, and we formed an interdisciplinary team comprising colleagues and experts from pathology and medicine. So with the help from everyone, including our colleagues in the ICU, our postdocs in the lab, and most importantly, our patients and their families, were able to collect multiple tissue samples from patients who succumbed to the disease. We subjected the samples to single nucleus sequencing, which is one arm, and the other half of the same sample to spatial profiling. So single nucleus sequencing has been transformative for us, especially for the tissues that we had to work on, but also having the ability for regions of interest, getting and 18,000 genes plus in situ with additional probes for SARS-CoV-2, so you could actually catch how much virus you have in that specific region and quantify the abundance. And we're very grateful with the support and access to the assay. So NanoString provided access to the assay about a year before becoming public, and that ability to be able to interrogate tissue with such a high plexity was a driver for this effort. And I would like to pause a little bit for those who are not familiar with the assay to just explain it, but also present some things that are key based on our experience. I stole these figures from NanoString website, so I put the logo there. I hope it's okay. So the process is quite straightforward. You can stain with 3 antibodies and DAPI your tissue. It really helps if you have serial sections with other staining or additional information as well. And then you can select up to 80, 90 regions of interest on your slide. These regions of interest are then photocleaved. You get your RNA with oligos, mentioning that specific location, and then you can either count or sequence. The assay that we use, which is the whole transcriptome atlas, is sequencing based only. So this is an example from the same website for ROI selection. I'm showing this since every assay has its tricks and its important details, and region of interest selection for the geomics, I think, can really give you things that maximize your research potential. So this is how this is an example from the website. These are different regions of interest from the same tissue, and this is you get thousands or 1,000 or 18,000 depending on the assay of genes for that region. So going back to the project, and I will come back a little bit to the strategy. I think the tactical components we can figure it out, but strategy can really derail or really maximize what you're doing. So along the way, our BIDMC effort joined a Boston-wide consortium to expand this cohort with exceptional colleagues from academic medical centers and the broad. So through this extensive consortium, we were able to collect more than 420 patient samples, 11 tissue types. 100 samples were processed for the original publications, and this effort still continues. We performed single cell and single nucleus sequencing per sample, as well as spatial proteomics and spatial transcriptomics using the geomics. And different teams have different approaches, which I think makes it even more interesting and more powerful. The original Atlas paper comprised results from 4 tissues. We focused extensively on lung since it's the organ with primary interest in COVID-19. And we were able not only to provide like a very detailed cellular map, but also to prioritize, and that is, I think, the power of this approach is to prioritize cell targets, not only cell-specific genes, but also specific cell states or cell populations, explaining a substantial part of the COVID-19 phenotype. As I mentioned, the focus was in lung, so it was for the ARDS lung phenotype, which could potentially be leveraged for really targeted therapeutics. Sami Farhi's team at the Broad used the geomics platform, and they followed the region of interest strategy based on pan-cytokeratin signal. So the selection and comparison between CK plus and CK minus regions really helped identify not only differentially gene expressed genes and the transcriptional programs that dictate these regions, but also by combining it with a concordant single cell / single nucleus data, you could identify which cell types and abundances per patient are there. You also have the viral compartment, so you can understand the viral quantity for these regions, and you can, for instance, see that you have an epithelial 81, 82 phenotype there with a higher viral abundance for some patients, while the pan-CK minus is mostly mesenchymal and endothelial component. Our group followed a complementary ROI selection strategy. So we formed a group that comprised hepatologists, pathologists, experimentalists, domain experts, computational biologists, and formed Zoom calls. Of course, Zoom, because it was also the beginning of the pandemic, but also it's easier for everybody to have a shared screen. And of course, with working with the technology developer at the same time, we jointly annotated and selected the regions of interest. So these calls, because the data were many and for many regions of interest and for many slides, they could be 5 or 6 hours long. But the process was roughly as follows. So we started with H&E. We had immunohistochemistry against SARS-CoV-2 spike protein, and then ISH using RNA scopes, a single molecule fish against SARS-CoV-2, ACE2, TMPRSS2, in order to identify viral presence in the tissue sections, and also the proteins that are the markers for viral effective loading on the cells, and then that guided the region of interest selection. And that is important to show that you can use circles, as we showed as we saw on the nanostring slide. You can use squares, rectangles, but you can also do freeform. There are other ways to do it, and I will show more on different organs. And for instance, this is a bronchial epithelium selection, and you only can select that. If your regions are usually 50 cells and up, I think officially it's 200 cells and above, but for us it works for 50 and above. You can get a very nice representation, and you get whole transcriptome for those. And viral scores were calculated and correlated with additional probes. So you have virus from the ISH, from the RNA scope, but you also have virus per region, which enabled us to do viral scores and identify localization and understand how virus correlates in situ with the phenotype that you see. And our team's region of interest selection strategy for lung, as I mentioned, focused on inflamed and normal appearing alveoli, which can be side by side, but they seem to have a dramatically different transcriptional program. So you see inflamed alveoli with red and normal appearing, at least, alveoli. These are very severely affected patients by COVID-19. And they can be side by side, but the transcriptional program can be different. But what's really cool is when you have that many genes to work with, you can do pathway analysis in situ. You can do your cellular types deconvolution in situ. And as we can see, this is an example of interferon gamma activity, and you can these are the same regions, so it's matched, and you can see how on the same lung sample the regions change, and how does this affect the gene expression for the same regions of the same patient or across patients. So on the single cell and single nucleus front, I will not dive into details today. I think more folks are interested to see the applications of the spatial, but I think it's very important to stress that how you analyze the data can really affect your capabilities. And there were amazing people on this consortium, like Bolly's team or other teams that really helped push the envelope further. So this is an example for cell bender that was just applied and gave us the ability to remove ambient RNA from the single nucleus sequencing, or other methods that were developed in-house, and enabled the quantification of the viral abundance per cell. So we see, for instance, that there is a myeloid compartment as a high viral content, and you can also understand the transcriptional programs. The team was able, I'll just move quickly. If there's more interest in the Q&A, I can go into more details. So the team was able to identify what cells are different in the healthy and the patients. Also identify cellular populations that are specific for COVID-19. And the two that really stood out was a pre-alveolar type 1 transitional state, PATS, which was previously mentioned in the literature of pulmonary fibrosis as 82 to 81 transitional cells. And they show defective differentiation and failure to replace damaged 81s, as well as an epithelial progenitor state first observed. It was the first time we observed it in humans, but it has been seen previously in H1N1 influenza affected mice, which was termed IPBLPs, which is intrapulmonary progenitor cells. And these progenitors are evoked to repair the epithelial barrier, but in fact, they exacerbate the loss of functional alveoli and hypoxia. So this is a graph from the paper showing how PATS and IPBLPs cellular states, which are completely absent in the healthy lung, how they affect, actually they signify failed attempts of the ARDS lung to repair and regenerate, further exacerbating the COVID-19 phenotype and hypoxia. So our team continued the investigation and generated spatial and single nucleus sequencing data from additional organs. We see here kidney, heart, spleen. And I again wanted to stress the importance of bringing people together around the same problem and owning it. So the strategy for region of interest selection was adapted to each organ. And we also use, so for instance, this is a region of interest selected free form here. Or another powerful approach is to use the markers and select only the positive cells or negative cells, or separate them per region of interest. So this example here, for instance, it could be, you can select your CD45 separately, and your pancytokeratin, or use the channels to select regions and sequence them separately. So I will use liver as an example for different types of analysis, but also an A to Z mature analysis pipeline. So for liver, our team's region of interest selection was mostly informed on liver architecture. We know that in liver, the oxygen gradient that is formed, you have high rich oxygen regions in the portal area, and access to oxygen and nutrients diminishes as you go to to the central vein. So this gradient dictates partially a liver cell function, and also enables us to split the lobular area into 3 zones: zone 1, zone 2, and zone 3 of the liver lobule. So liver is an organ where spatial structure and architecture really play a very big role on function. So that was very key to guide our region of interest selection. And also we used CD45, so we got region CD45 plus from the same ROI, and then CD45 minus. So this combined team effort also enabled us to generate in collaboration with MGH, the Broad, New York Presbyterian Hospital, and Brigham and Women's, to generate more than 90,000 nuclei, which for human liver, it's an incredible effort. And single nucleus sequencing, actually, all those are single nucleus sequencing, enables a very accurate representation of all the cell types, and they reflect how the actual liver is. So more than half are hepatocytes. And if you go to amazing and leading publications that use single cell, you know that for hepatocytes, there is an overrepresentation of the non-parenchymal compartment when you're doing single cell sequencing, because liver hepatocytes actually are quite difficult to capture with single cell, but with single nucleus, this is actually solved. So the very high number of nuclei enabled us to characterize in high resolution the function of hepatocytes, identifying numerous affected cells exhibiting stress, apoptotic, or acute response phenotypes. And we were also able to characterize the immune, endothelial, mesenchymal cell compartments in high detail. I will just very quickly drive through those, since I think we would want to spend more time on the spatial, but it's important to capture your cell types accurately, even rare cell types, in high granularity. We see here the cholangiocyte compartment, which is usually quite rare in many different data sets, because that enables you then to understand what you're seeing. So many new techniques we had to implement on the single cell component. Normalization that enables you to get normalized counts, so you can use normalized counts in your trajectory analysis, or as we see here, an example of identifying viral reads in higher resolution than we could before, just because the liver is not as affected as the lung, so we had to increase at the same time sensitivity, but also the signal to noise ratio, in order to be able to get viral reads from the single cells, which were then later corroborated with qPCR data and with RNA-scope as well for the same patients. So having SARS-CoV-2 abundances per cell is I think it's quite important, because first of all, you can show which cell types can be affected. You can understand the correlation for liver, because for lung, we definitely knew, and there were a lot of questions whether hepatocytes, for instance, can get infected or not, or which compartments get more infected or not, and how that correlates with ACE2 and TMPRSS2 and so forth. And okay, so that is definitely important. But the other thing is that you can identify the cells that are infected, and then you can identify and overlay transcriptional programs for these cells and start understanding what the infected cells are doing, what signaling molecules they're expressing, how they affect their microenvironment, and so on. So, of course, having spatial data and knowing also in each of those regions where the infected cells are or not, and as we mentioned, with regions of interest that really dictate liver function, as well as the ability of increasing the granularity using the markers and sequencing only specific regions, you can get very highly localized information and understand which cells localize and co-localize. And as I mentioned, data ingestion for spatial is very important. Just as, just for the liver project, just for this effort, within the 6-7 months from the Atlas project to this, the team had to design and implement many new methods to maximize the utility of the geomics WTA, new raw data normalization from scratch, 2 new batch effect correction methods, pathway enrichment and activations in situ, cell type deconvolution. It's many of the methods that are made for bulk or for other assays, they simply do not provide the accuracy that you want for a new assay, and everything sometimes they have to be optimized or done from scratch. So Jared from our group spearheaded the effort to normalize efficiently the WTA data, and we see here there is a slide. So I mean, it's quite evident there. So there are 2 patients on the slide, and we see that there is a slide-to-slide technical noise. And if you zoom into a single slide, you see that patients also segregate. So there is a patient-to-patient noise. And if you do standardized methods, so this is the original, if you do patient adjustment or slide adjustment or both, you still cannot capture that clearly, very fine information like zonation. Here we see one of the methods, BSSR for short, that Jared developed, and we can see that we get zone 1, 2, and 3 following the normalization, and not only brings patients together, but you can still retrieve a very refined biological signal, such as hepatocyte specialization, because the populations in these regions are practically almost identical. It's just what they're doing that changes. So the method was redesigned during the past few months and extended by Nicholas from our group. It enabled us to acquire a very clean signal also for zone 2 regions. As you see, they're collapsed here, and enabled us to also segregate those, and which is the first time for human liver data, because zone 2 is not a real, is a transition between the 2 states. So having, as we mentioned, the single nucleus data at the same time and spatial data from the same samples enabled us to register the single cell clusters in situ and maximize granularity. This is an example where this is a specific cluster of cholangiocytes, and we can see in situ using the single cell data where those cholangiocytes are, and that in some lobules they're 100% periportal, but in but in other lobules we see also that they extend to zone 1, and this has a lot of implications for depending on the patient and the phenotype. So Jared from our group and Priya from Wing Heights Lab, they developed also a method specifically for the WTA for pathway activity using in situ spatial transcriptomics, which enabled us to identify per zone or per region which pathways are active, goes way beyond enrichment. I'm more than happy to discuss about this further. I think it has been a big driver on our annotation process. But also, okay, you can understand, because you know which regions are affected by the virus, which regions are not, to understand what is the functional signature for those. And this is, of course, ongoing work. And I would like to close mentioning the most important, at least for me, take-home message, that the integration of these 2 technologies on the same patient samples is what's bringing this together. you can get your granularity and your like maturity from the single cell, but you cannot go without the spatial component. And that I think is key. There will be a point, again, this is another quote, and it might not age well, but I think this one will definitely age well. There will be a time where spatial will not need that much, having the additional component of single cell at the same time, because you will have the granularity and the capture rate, and the methods will be more mature. But for now, if you want to get there, having both at the same time really helps. So closing this work was the result of a collaboration of many talented people. It really takes a village. It's an extreme privilege today to be able to show even a small part of this work. And also I would like to provide an example of team science. So this is a team science effort, but I think every spatial project, if we want to maximize it, is team science. For the liver paper, in order to enable the domain experts and all the team to work on the same data easily, our team developed collaborative apps for the spatial, for the single cell. We had content curators curating the literature and manually selecting pathways from the literature or from databases, multiple domain expert meetings, countless, and then follow up everything that was identified on the single cell, the high throughput, go back to stains or reanalyze public data to see if that is real or not. As an anecdote, as an example, while Nicholas was developing the method for normalization, there was a specific patient that seemed to need a little bit stronger modeling to iron out. And of course, as the bioinformatician's reflex is, yes, let's increase a little bit the stringency and this signal will go away. But having domain experts and the ability to go to do in situ work, we worked with the chief of hepatopathology of BI, and with spatial stains, we saw that no, this is real. It's real signal. It shouldn't be ironed out, and we're happy that the method is sensitive enough to pick it up. There was a very strong phenotype for that patient, which we followed up with pathology. So special thanks to the members of our lab and the BIDMC team for the tremendously long hours and the incredible pace during the beginning of the pandemic, and nonstop since then. The technology developer for early access and tremendous support. And of course, once more to thank our hosts today for the very kind invitation, and for you to for adding one more Zoom meeting on your Friday schedule, and would be thrilled to take any questions. I would like to close with this plug slide. We have multiple positions open, from leadership to trainees. So if you want to dive head on, in this amazingly expanding field of spatial, please feel free to reach out. And thank you.
Thank you, Dr. Vlachos, for this great presentation. It's impressive that you have all these spatial technologies in just one place.
Yeah, it's and we're also, it opens up questions how you should optimally integrate, because initially we were saying let's select the right one, but maybe some questions need more, and maybe some questions can be answered by bringing them together to capture multiple aspects. It's it still is nascent. So
nice. And if the attendees have questions, please feel free to unmute yourself and ask, or post on the chat box so we can ask. So you talked, in the meantime, maybe I can ask a few questions. You talked about normalizations, right? So the capture efficiency of all these machines and technologies are different. Do you also take into account those as well? Capture efficiencies and the readout differences?
So it depends on the technology, and I think you're bringing two important aspects on the same question. So first of all, even if the technologies might seem different or might seem similar, for instance, in the in situ hybridization field, there will be multiple, capture rate, accuracy, they will be different, and there is no way to standardize yet, and there is no way to actually quantify. So many things happen And for us, at least for the WTA, we had to rely a lot on our domain expertise. We happen to work on liver quite a lot, and we have analyzed many public single cell and bulk data sets, and also working with our hepatologists and pathologists. It's very difficult because there is no standardization. There's no way to actually quantify it. It's still quite early. We were relying on the public normalization methods initially, but we saw that there was a lot of signal lost. So we went, to the drawing board from scratch, and we started working with the data until we understood how the assay works to create a normalization that is sensitive enough yet robust enough at the same time.
Great. Yes, exactly. Robust enough, right? And also, I just looked at your also technology group website. Are you guys going to also collaborate with different institutes, or this is mostly Boston based?
So this is open. No, this is open to it. And our mandate is actually how many we have, and we're evaluated on how many we have helped, not only internally but also externally, be it academics or startups or the industry. If you want spatial to be on the front line, you have to open to the community. Otherwise, it's just the lab that has access to the instrument, and you close your door, and it's good for your science. But if you want spatial to be revolutionary and transformative, is providing access to everyone, and it's first in, first out.
Great. And let's say if they want to get some preliminary data, could they work with your group or unit?
Yeah, of course. I mean, it's a facility. It's service. So there are opportunities to collaborate with the lab. That is one thing, but you don't have to. You can work directly with the unit, and then you will work with. We have specialists in there, and they will work with you, guide you, match your question to the instrument, or if you already know, just make sure that it is the right assay for you. And then yes, that's it. Some assays are easy for pilot data. Other assays are usually, if you have to design a new probe set from scratch, or you have to design a new antibody panel from scratch, that might be a bigger time investment for a pilot. But we have already pre-set or pre-made versions, and if they're compatible, that's also an easy way to start.
Great. And your also team includes the bioinformatics custom algorithm development too?
So this is mostly how to analyze spatial data there is no best practice yet. And for many of the questions, there are not even methods yet available. And for many, you just develop as you go. So this is where collaboration again is important. The unit can collaborate with our lab or Winhide's lab or the other labs that are supporting it for new methods development. We avoid doing methods development as a facility, as a core, because it's open-ended, but we can bring the labs close to the question to develop, for instance, a new method. But also we're working closely. We don't consider the vendors as they just sell the instrument. We consider it as a collaboration and as the technology development. So we have a two-way street with their R&D, and when they have progress or when we have progress, it's a two-way street in order to make those assays more applicable for anyone. So I think it's a hybrid model. For straightforward analysis, just the STU can tackle it or can connect with bioinformatics cores or your bioinformatics core, for instance. But if it's open-ended technology development, the STU can connect with labs to develop a new way to approach the data.
Great. Now, are there any questions from our participants? In the meantime, for COVID studies, you talked about a new couple of initiatives for variants and all that, right? More getting into the granular details of also COVID variants. Do you guys have any efforts identifying new variants and new developments of possible RNA therapeutics, right?
So for spatial, or so mostly we were working with these approaches for bulk. We are now like shyly bringing those to spatial. Our TCR work, we're bringing it into 2 assays currently for spatial. We have more years working with bulk, so it's relatively more straightforward if we have an idea how to implement it. Many times with spatial, we have to work with a technology developer and try to also change the prep or work with the assay in order to increase granularity there. But yes, there is definitely potential for bulk, coding, non-coding mutations, their effects. We have projects dedicated for that. For spatial, the sequencing-based assays are quite powerful because you can get more than you expect based on sequence. And the assays that have very high resolution, like the single molecule fish, and where you can go subcellular, you can get some very, very nice questions answered for translation. If your RNA is in the plasmic reticulum, you can get information about your long-encoding RNAs if they're in the nucleus. You can get a lot of things that the assay was not meant to address, but it's free information for you there.
Exactly. And also for the technical concerns, again, the FFP versus fresh frozen directions, right? So when people are interested in either or both, right, which one would you recommend? I mean, is FFP really a loss of a lot of antigens and RNA molecules, or they're still good to capture, let's say, the phenotypes in multiple disease models?
Yeah, that's a good question. So our department, I don't know, does 50,000 blocks a year, or I have no idea, maybe more. Having the ability of interrogating those archival samples is gold, right? So FFPE is, and with FFPE, we have the ability to do tissue microarrays. We can put 40, 20, 160 patients on the same slide, which cuts down costs. You can very quickly screen. So there are a lot of benefits on FFPE. On the other hand, I mean, if you work with RNA, you know that FFPE has many challenges. We have worked FFPE for the protein-based, like the codex, for the Polaris, it's not a problem. For RNA work, for the nanostring, actually almost everything we did was FFPE, and it works great. We don't see any difference. With Visium, we're working again on the FFPE. For Merscope, we haven't piloted it yet. So for Merscope, that would have been a very nice protocol to iron out. We're in very close communication with the technology developer, with FizGen, to set this up as well. But yes, I think the best option would be to have both protocols equally robust. But FFPE has its own challenges. So capture-based, when you have probes, it really helps. That's why, for instance, Visium and the genomics are actually quite robust, and the data look good.
And we have one question from Bjorn. And do you yourself, or do you have your pathologists select region of interests? Yeah,
so this is the question I was expecting, and I think domain expertise can for assays like this. But even if you do Visium, right, or even if you do something you're hypothesis free, how you make sense of what you got, you need domain expertise. You need someone to have the ability to understand tissue, that specific organ or tissue that you're interested in, and the very refined nuances. I'm very fortunate. I'm Department of Pathology, and so it means we have access to brilliant people around. Plus, for liver, we had 3 hepatologists on the program, plus a hepatopathologist. So each one of those made a big difference. So having access to domain expertise, that's why I think spatial is the archetypical experiment or investigation that is truly team science. you need your histologist, and you need a really good histologist to prepare your samples. You need really you need good hands on the bench to prepare your assays and or to sequence. Your bioinformatics, it's still research in terms of bioinformatics analysis, so people should be really good at what they're doing, and you might need new technology, new like methods development, and then knowing the organ well, you cannot do without. So you need this circle of trust around the single project where everybody feels ownership to help.
Nice. And Natalia has a question. Just as a ballpark range, what would be the price charged for 5 to 10 samples for an outside order from an academic university?
So that's a question that it's very common, and you go in the elevator and say, oh, Ioannis, oh, by the way, I have 10 samples. What can I do because I have a pilot, or how quickly because there is an NIH call, or how much it would cost. Shoot us an email. It's usually more complicated than this, which technology, which question, what type of samples. Yeah, just shoot us an email, the website, and we will connect you with one of our, with a scientist that can really guide you through the process. And that is the important thing, if you only have a hammer, then it's a different question. But the important thing is for the research problem you're facing to be solved, actually.
Great. Are there any other questions? If not, let's thank to the speaker, and I'd like to use this clapping sign again. Thank you. This was fantastic. Vlachos. And with this, I think I'd like to announce the next week's speaker, Dr. Lu Wei from Caltech Chemistry, and she's going to talk about high-resolution multiplex chemical imaging in single cells and tissues. And I think at this point, I think we're hopefully ready to wrap up. And thank you again, Dr. Vlachos, for your fantastic presentation.
Thank you so much.
Bye-bye. Everyone, have a nice weekend, and hope to see you guys next week. Bye-bye for now.